reproduction in lower and higher plants, you have studied about how male and female gametes are formed, then asexual and sexual reproduction as two basic types, pollination, then adaptations to favor certain type of pollination, all these points. Now today we will do the next part in the lesson, that is actual process of fertilization and what happens after fertilization, that is post fertilization events. Okay. Now let us first do process of fertilization. For that I will quickly revise that pollen grains germinate on stigma, and every pollen grain after germination produces a pollen tube carrying two non-motile male gametes. This is what you have studied earlier. I have also told you that these two male gametes, they are non-motile. So if they have to reach a female gamete, their movement takes place along with the elongation of pollen tube, and this is called siphonogamy. Okay, I repeat, siphonogamy is movement of two male gametes along with pollen tube from stigma to ovule where there is female gamete or egg cell present. Now, in angiosperms, the process of fertilization is very peculiar. That means it is double fertilization. Remember that angiosperms are the only group of plants where double fertilization is seen. Now, let us see what is this double fertilization. In fertilization process, you can see here I have drawn a diagram. This is angiospermic ovule, which you have already studied, anatropous or inverted ovule. Near micropyle, you can see egg cell. Near chalaza, antipodals, and roughly in the center of the embryo sac, there is secondary nucleus. Now you can see this is the pollen tube, and pollen tube carries two male gametes. In the process of fertilization, these two male gametes are used up, and event of fertilization takes place twice. That is why it is double fertilization. I am repeating, in fertilization in angiosperms, two male gametes are used, and that is why event of fertilization takes place twice, and therefore it is double fertilization. Okay, now let us see what happens actually in this process. Now this pollen tube, if it enters through micropyle, right now in this diagram you can see pollen tube entering the ovule through micropyle, then it is called porogamy. Porogamy means entry of pollen tube into ovule through micropyle. Now suppose if pollen tube enters from top, that is from chalaza side, then it is chalazogamy. And if pollen tube enters from lateral side, piercing integuments, then it is mesogamy. Okay, so there are three terminologies: porogamy, entry of pollen tube into ovule through micropyle. If pollen tube enters from chalaza, it is chalazogamy, and if pollen tube enters from integuments, then it is mesogamy. But remember students, porogamy is very common in angiosperms. Okay, so you have studied these different terms as siphonogamy, porogamy, chalazogamy and mesogamy. Now let us assume that pollen tube has entered through micropyle. Now what will happen? This pollen tube, it starts growing towards embryo sac and it enters the embryo sac. Now after entering embryo sac, you can see here there are two synergids. From one of the synergids, pollen tube absorbs water. Now because of water absorption, pollen tube swells and it bursts. It bursts to release both the male gametes. So now what will be the situation? Inside the embryo sac, there will be two male gametes released. Now, one male gamete unites with egg, okay, and that is called syngamy. Syn means union or fusion, and gamy stands for gametes. So, union of gametes. So, here egg, that is female gamete, and male gamete, they unite with each other to form a zygote. Now here, see, both the gametes are haploid, and that is why zygote which is produced is a diploid structure or a cell, and zygote will be beginning of the next generation. So a diploid zygote is produced after syngamy. So this is how one male gamete is used. Let's see what happens to the second male gamete. Now second male gamete unites with this secondary nucleus. Okay. You know that secondary nucleus is already binucleate, that is diploid 2n. So, secondary nucleus unites with the second male gamete. So, here you can see diploid secondary nucleus uniting with haploid male gamete, and that is why a triploid 3n structure is formed. It is called primary endosperm nucleus. Short form is PEN. It's a triploid structure, and this particular event is called triple fusion. Okay, because here a triploid cell is formed after this fusion, that is why it is triple fusion. So here you can see two male gametes are used up in fertilization, and that is why it is a double fertilization event. Now you can open page number 10 of your textbook. Now we will do significance of double fertilization. Now it is a unique feature of angiosperms. I have told you in plant kingdom only angiosperms show this particular feature. It ensures that parent plant invests a seed with a food store only if the egg is fertilized. So here this is the way by which a seed is formed. then diploid zygote develops into embryo. Now this zygote, if you allow to grow, zygote is unicellular. Afterwards what happens? A unicellular structure becomes multicellular by divisions, and that is called embryo. So this embryo is formed, and ultimately this embryo will give rise to the new plant. So because of double fertilization, there is embryo formation. Now you can see here both the male gametes are used, and in that primary endosperm nucleus is formed. Now this primary endosperm nucleus, initially it is just one cell with triploid structure. Now primary endosperm nucleus also divides by mitosis and forms a structure called endosperm. Endosperm provides food to embryo. So that is why formation of primary endosperm nucleus is very necessary to provide nutrition to developing embryo. Then next is diploid condition is restored. That is when zygote is formed. Zygote is diploid. Zygote will later give rise to diploid embryo, and that's how the new plant which is formed, that's a sporophyte, is also diploid in nature. So this is how that diploid chromosome number is restored, and polyembryony is avoided. Polyembryony means many embryos growing at a time. If multiple embryos are there growing, then they may not get sufficient nutrition, and that will affect their development. That is why in angiosperms, usually single embryo develops at a time inside the ovule, so that single embryo gets better nutrition, healthy embryo is formed, and naturally next generation will be also healthy. So here polyembryony is avoided. Actually, if you see this diagram, embryo is diploid because zygote develops into embryo. So remember students, if there is no double fertilization, secondary nucleus which is already diploid may form second embryo. First embryo is by normal fusion, egg and male gamete are fusing to form a zygote which develops into one embryo. Second embryo may develop from secondary nucleus which is already binucleate, and third embryo may be formed from second male gamete and synergy. And this is how multiple embryos may form inside the embryo sac. And to avoid this, there should be double fertilization which will prevent polyembryony. Okay, so this is double fertilization process. Now from exam point of view, process is important. Then all these terminologies which you have studied just now are important. Practice drawing diagram which is given in the book, plus significance or importance of double fertilization. Okay, so this part from double fertilization is important. Now let us do next part. How does primary endosperm nucleus develops into endosperm, that is endosperm formation? endosperm formation is nuclear endosperm. Okay. Now in nuclear endosperm type, we'll begin with primary endosperm nucleus cell, which is produced after double fertilization. Now nucleus of this cell divides by mitosis. Okay. Many divisions take place. After few nuclear divisions, many nuclei will be formed. So these are going to be free nuclei. Remember here in this particular type of division, only nucleus is dividing. Initially there is no cytoplasm division. So what will happen is, after repeated nuclear divisions, there will be many free nuclei produced. And once the sufficient number of nuclei are formed, you will see that cell wall is produced. So every single nucleus is bounded by a wall. And now you get a multicellular structure. This is called nuclear endosperm. So initially only nuclear divisions take place, few nuclei are formed, and then there is cytoplasm division. Okay. Now second type we will see, it is cellular type. Now in cellular type, what you find is this triploid primary endosperm nucleus, it divides and division of nucleus is immediately followed by cytoplasm division. Okay, and then you get a multicellular structure formed. So here every time what happens is nuclear division is followed by cytoplasm division. And the last type is helobial. And in helobial type, we start with primary endosperm nucleus. Now this primary endosperm nucleus divides into two nuclei. Two nuclei separated by a wall. So you can see here two cells are formed. Now in both the cells you see free nuclear divisions, like the first type, that is many nuclei will be formed by mitosis, and then every nucleus gets a wall. So this is how a helobial endosperm is formed. So these are three types of endosperm formation, and this endosperm will remain inside and will provide nutrition to the developing embryo. So finally what will happen is, once the embryo develops into a matured one, this endosperm is usually consumed by embryo. So in most of the cases, endosperm is absent at maturity. There are examples where endosperm still persists and they become endospermic seed, and if there is no endosperm in the seed, it becomes a non-endospermic seed. Okay. Now other things, that is embryo development, we will do next time.